Hello, everyone. Thank you all for tuning into this talk. My name is Katie Gorick, and I'll be speaking about some recent work at UVA investigating the use of focused ultrasound-mediated blood-brain barrier disruption to facilitate delivery of mitochondria to infarcted brain tissue following an ischemic stroke. As a little bit of background, strokes are one of the leading causes of death and permanent disability worldwide. Roughly 800,000 Americans suffer from stroke each year, and nearly 90% of these are classified as ischemic stroke, which occurs when the blood vessel in the brain is obstructed, most often by a blood clot, which cuts off the circulation to part of the brain tissue. We can recapitulate many features of human ischemic stroke with a mouse model of transient middle cerebral artery occlusion, in which a suture is fed through the internal carotid artery to the branch point of the middle cerebral artery and left in place for 1 to 2 hours to induce ischemia before being removed to allow for reperfusion. In recent years, there's been increasing interest in the potential application of mitochondrial transplantation to promote recovery of ischemic tissue. Clinical trials are already underway investigating the efficacy of this approach in the context of pediatric cardiac ischemia. In this approach, mitochondria are harvested from the patient's own skeletal muscle, purified, and then injected into the infarcted region of the heart. This recent trial shown on this slide has found that the mitochondrial transplantation improves ventricular function after ischemia reperfusion injury. With this in mind, we wondered if the same improvements might be observed following mitochondrial transplantation in ischemic brain tissue. So following a 1-hour middle cerebral artery occlusion, mice received an intra-arterial injection of either mitochondria harvested from their own skeletal muscle or a vehicle control. In panels A and B, you can see tetrazolium chloride staining of coronal slices of mouse brain tissue. With this TTC staining, healthy brain tissue appears red and the infarct region appears white. I've also denoted the infarct regions with blue circles. As you can see, the intra-arterial mitochondrial delivery results in a significant decrease in infarct volume 24 hours after stroke. While the blood-brain barrier is already compromised in the context of ischemic stroke, mitochondria can be as large as multiple microns in diameter, and we may not be achieving as much delivery as could potentially be possible. We therefore sought to determine whether FUS-mediated BBB disruption could enhance delivery of the exogenous mitochondria. We first needed to determine whether the infarct could be appropriately visualized and targeted with FUS. As you can see here in the T2 image in panel A, the edema from the ischemic injury is hyperintense and can be effectively targeted with our MR-guided FUS system. And as we can see in the contrast-enhanced T1 images in panels B and C, we're able to achieve sufficient microbubble permeation even in the dysfunctional vasculature of the infarct region to get significant contrast extravasation in the stroke tissue, which we hope will correspond with increased mitochondrial delivery. Here we've injected Evans blue dye intra-arterially into the mice with and without MCAO surgery and mice with and without FUS-mediated BBB opening. As you can see from these TTC images, while there is some accumulation of blue dye in the stroke brains without FUS, which will be panel C, due to the leaky and dysfunctional vasculature, the dye accumulation is greatly enhanced when we use FUS and microbubbles to open the BBB further in panel D. So we next wanted to see if the same phenomenon would be observed with intravascular mitochondrial injections. This is a brief overview of our experimental methods. We used MR-guided focused ultrasound to transiently open the BBB in the ischemic brain tissue, and then delivered fluorescently tagged mitochondria from the animal's own skeletal muscle tissue intra-arterially via the same catheter that we used to deliver the microbubbles. Animals were sacrificed 24 hours later for analysis. In these images, we're seeing DAPI in blue and fluorescently labeled mitochondria in red. As we might have expected from the Evans blue experiment, we see little to no mitochondrial accumulation in the healthy brain tissue, some accumulation in the stroke brain, and robust accumulation in the stroke brain, which has been further treated with FUS and microbubbles to open the BBB. So clearly this FUS-mediated BBB disruption is useful in getting more of the intra-arterial mitochondria out into the surrounding brain tissue. This was further verified by high-power confocal imaging of the mitochondria relative to the vasculature, which we stained here with fluorescent green CD31 antibodies. And as you can see from the quantification on the right, while the intravascular levels of the mitochondria were similar across treatment conditions, there was a significant increase in the amount of extravascular mitochondria following the FUS-mediated BBB disruption. Our next steps for this study, which is currently in revision at iScience, include quantification of the infarct size after the FUS-mediated mitochondrial delivery, as well as after a direct ATP injection. And this is to determine whether the improvement that we see in infarct size is purely due to increased ATP availability or if there's another factor of the mitochondria which might be beneficial. We also want to investigate other metrics of cell survival and function, as well as long-term behavioral functional improvements, such as rotor-rod performance testing, to see if behavioral effects of the stroke can be attenuated or even reversed with mitochondrial transplantation. I'd like to thank my lab, our collaborators, and our funding sources, and thank you so much for your attention.